Welcome everybody to another Merit Laboratory Partners tutorial. This video segment is going to talk about pharmacogenetic testing. In fact, we'll be doing that in the next two videos. In the last video series, the one on basic genetics, we talked about the fact that protein is derived from DNA, and specifically the sequence of DNA has a big impact on the sequence of protein. Now, that sequence of DNA we called genotype. We also mentioned single nucleotide polymorphisms. These are little DNA variants that can have a big impact on protein sequence and therefore on the function of protein. I also suggested how researchers can often find very important clinical correlations between a particular DNA sequence and protein function. And let's just look at a more specific example now. Let's pretend for a moment that we're interested in gene ABC, which we know metabolizes drug X. If we start looking at DNA sequence, we might find where individuals who have two wild-type copies of gene ABC, they're able to benefit from drug X. However, if they have one wild-type copy and one mutant copy or one variant copy, it could be that they only benefit marginally from drug X. But if they have two copies of the mutant DNA sequence, and therefore two copies of a mutant protein, then maybe they don't benefit at all from drug X. So this is a very generic example, but it's the kind of information that we can get from looking at DNA sequence. Let's then talk about pharmacogenetic testing itself. So pharmacogenetic testing, also known as PGX testing, is just a diagnostic tool that allows us to test specific DNA sequence variations in patients. Using that information, we can then leverage the latest clinical research to make predictions about how that patient is likely to respond to specific drugs. Well, in humans as well as in other mammals, there is a family of genes that plays a really big role in metabolizing pharmaceutical drugs. And actually not just pharmaceutical drugs, but really any xenobiotic. And the products of these genes, what they do is they either convert inactive prodrugs into their active compounds, or they break down the active drugs into their inactive metabolites. Generally, it's the former. Collectively, these genes are known as cytochrome P450 genes, or simply CYPs. And there are a lot of these genes. In fact, there are over 50 of these identified in humans. And every patient that we look at is going to have a specific metabolizer phenotype for each of these CYP genes. And of course, that phenotype is based upon the individual's genotype. Well, here are several phenotype possibilities. A person could be an ultra or rapid metabolizer, meaning that they're going to take that prodrug that's being introduced into the patient's system and rapidly convert it into its active drug. They may be a normal metabolizer, meaning they're going to make that conversion at a fairly normal rate. They may be an intermediate metabolizer, which is somewhere between very slow and normal, or they may be a poor metabolizer, so that conversion is done at a very slow rate, or sometimes not at all. And because different genes, different ones of these CYP genes, metabolize different drugs, a patient can actually have a rapid metabolizer phenotype for some drugs, but a normal or poor metabolizer for others. And here's an example of 7 commonly tested CYP genes. This individual, this patient, for CYP1A2, they're a normal metabolizer. But for CYP2D6, they're a rapid metabolizer, and it's important to know the metabolizer status for each of these genes, and that's why we test them individually. Now what we're seeing here are 3 commonly prescribed drugs: clopidogrel, also known as Plavix, trazodone, and warfarin. And you can see that each of these is metabolized by different genes. And in fact, in the case of warfarin, we actually have to look at a metabolizer gene and a drug target gene in order to fully understand the metabolizer status. But this just shows you that we have to look at multiple genes in order to make predictions for multiple drugs in any given patient. What then is pharmacogenetic testing ultimately telling us about a patient? Well, based upon that individual's alleles for each of the genes we test, we can then make predictions about whether or not a drug will be effective or ineffective in a given patient, whether the drug may be required at a low dose or a high dose or maybe something in between, and potentially whether a drug is going to be harmless or toxic. And obviously it's that last one that's really, really important. We want to identify drugs that a patient absolutely should not be on, and PGX testing can often help us do that. And our lab in particular tests patients for dozens of well-characterized DNA variants that are known to affect how genes either metabolize or transport many drugs that are commonly prescribed. And so when we make specific recommendations to the physician, these are the things that we're basically telling them. Are the drugs likely to prove effective? What drugs maybe should the doctor avoid? What dosages should the physician think about trying first? And finally, are there any known drug-drug interactions that the physician should be aware of? Ultimately, the main goal of pharmacogenetic testing is tailoring a patient's prescription drug regimen to his or her genetic makeup. And this is really the essence of what we call personalized medicine. Now, in the next video, I'll expand on this whole concept of personalized medicine. I'll talk a little bit about the specific genes that we test, how we do it, and what the reports look like. So please join me for that.